All right, we're here in Simplify Science Selects lecture over DNA complexities. Let's begin by talking about some important terminology. So we use this phrase, the central dogma of molecular biology, which is a very grandiose phrase, and it's a very, kind of big word. It comes back from the 50s, but it encapsulates this idea that's pretty important. And the idea is this idea of one direction. Not the band, the single flow of information from DNA into the RNA, then into the proteins. And that's the way that information goes, from DNA to RNA to proteins. We do not see information that's in a protein be converted back to RNA. We never see information go backwards. We never expect to. It's not the cycle. And so this is a really important idea, and what happens is that the one direction that information is in the DNA, that's where it's stored. And then it moves into the RNA, and it goes from the RNA to an unzip in the proteins. So we have replication is our first big word here, followed by transcription, and then into translation. So those are the three different processes we see. So replication is the backups. It's how we make backups of our DNA. So an enzyme goes and separates the DNA from a double helix into two single helixes. So we have two half pieces of DNA. Then in step 2, some free-floating extra nucleotides pair up with all those exposed bases. And so where we had two single sides of DNA, all this stuff starts sticking to it. And so this is done by an enzyme called DNA polymerase. That's what bonds the nucleotides and sticks them in there. So DNA polymerase kind of forms the other half of our broken molecules. And next thing you know, we end up with two new molecules, because we had one side of the original molecule that's been all patched up with new stuff to make the other side complete. And then we have the other side of the original molecule that's been all patched up again to make it complete. So that's why we use the word semi-conservative. So in replication, we start with one piece of DNA and we end with two. But we can't point at my two DNAs and say, this is the old one, this is the new one, because each one has half of the original molecule, and the other side of it has been newly built. So it's semi-conservative. The reason replication is important is because it makes sure that we can make backups of the information, so that every cell will always have a set of the instructions. It's very fast and it's very accurate. It's accurate because of the base pairing rules. DNA, your cells are replicating DNA so often that in your body somewhere right now, DNA is currently being replicated. It happens a ridiculously large number of times throughout our life. The second process is transcription. So transcription is one of the flow of information where we go from DNA to RNA. Now, why would we ever want to do that? Why would we ever want to do transcription? Well, because DNA stays in the nucleus and DNA never leaves the nucleus, because the nucleus is safe and we can't risk hurting our original information, the DNA. So anytime we need to use it, we transcribe the information over to RNA, and we send out RNA to do all the dirty work, because RNA can be destroyed because we still have the original. All right, so that's the why. Let's talk about how it happens. So first, this RNA polymerase starts this whole thing. So there's a little site called the start site. It's not a very creative name, but it is very descriptive. And it unwinds just a chunk of it. You can see on the left and the right that I still have the double helix, but I kind of unwind and open up this section in the middle. So from here, what I is inside the nucleus, where the DNA is at, I start building just a single chain of mRNA. Now, this is very similar to replication, except RNA, the molecule I'm building here, will stay single-stranded. It never gets the other side on it. The way we accomplish this is one of the nucleotides gets switched out. In DNA, we had A pairing with T and C with G. RNA has the same CG pair, but U takes the place of T. U stands for uracil. So the adenine to uracil pairing in RNA keeps it so it'll be a single strand. It's part of the geometry of it. So you can see that single strand in red there in the middle. That big shaded spot would be the RNA polymerase enzyme that's facilitating this reaction. We're building that piece. Once we get to the stop site and we've made one complete message that's been transcribed, a gene, we've transcribed the information from DNA to RNA. The RNA strand will separate, and then the DNA will be wound back up and closed and re-helixed, re-double helixed. I'm not sure it's a verb, but you know what I mean. So this RNA molecule that's single-stranded has now been processed, shipped out, ready to go. It's a single strand. The fact that it's so small and skinny is what allows it to move out and through the nuclear envelope, out of the nucleus, into the cytoplasm of the cell, the outside parts. And then it can be used there, and that's going to happen in translation. This is kind of a cool picture here. This picture was taken by a TEM. That's a tunneling electron microscope. Really cool piece of technology. It allows us to get and see inside of things. It only lets us see in black and white, but this image is a real picture taken using electrons instead of light, but it's at 13,000 times magnification, which is incredible. So we're looking at DNA. So you can see that's labeled in blue there, those single, like what looks like a single strand, which is actually a tightly wound double helix. We're just not zoomed in quite enough even to see that. You can see these other parts that have been unwound, and there's these little tendrils coming off of them. Those are strands of mRNA. There's lots of them happening simultaneously here. All these different RNAs being formed. You have to remember that our cell is really good at multitasking. It doesn't just do one and then wait for that to be done to start the next. So these are all different RNAs. So you can see some parts of it are wound up, just as DNA. Other parts have been opened up, and RNA polymerase is starting to make strands. So what is the central dogma? It's that idea that information only flows one way. It's not a back and forth conversation. It's a single flow. Information is in the DNA, then it gets transferred to the RNA. That's the direction. We never make DNA from the RNA. That's the wrong direction. That doesn't happen. We make RNA based on what the DNA says, but never the other way. What are the similarities between replication and transcription? So we'll get a couple here. Well, they both involve opening up the DNA molecule. They both involve moving information to another nucleic acid. They both involve creating a new molecule. Differences are that replication is making DNA and transcription is making RNA. The difference is that replication ends up with two new DNA molecules, and that transcription leaves my DNA how it found it and just makes an RNA molecule that leaves. What might scientists learn about genes by studying RNA? Well, if we understand the central dogma and if we understand the code, we can learn anything that we need to about the DNA gene by learning about the RNA gene. And in fact, the RNA would tell us where it's starting and stopping. Now, this piece of RNA is very specific to this very specific task, so it won't show us the whole picture, but it'll show us in this specific example, what is that gene going and doing? What's being encoded? What information is being transferred? And the last, translation. So in translation, we use the same word because it's the same as translating English to Spanish. We're moving from one language to another. In DNA and RNA, we were speaking the same language. We had some different letters, like in DNA we used A, G, C, and T, and then in RNA we used A, G, C, and U, but it was still the same language. Proteins speak an entirely different language. They don't have these bases or nucleotides. Instead, they have amino acids. It's still the same idea. The sequence is what's important. But in the English language, the sequence of A, B, C, D, F, G, H, I, J, K, L, M, N, P, Q, R, S, T, that is the information. In DNA, the sequence of A, G, C, and T, that's the information. In RNA, A, G, C, U, that's the information. In proteins, it's a chain of these 20 different amino acids. That's the information there. So information is always encoded in a sequence, but the question is, what language are we using to communicate that information?